Okay, today we'll be going over the process of meiosis. So the purpose is to create four genetically different cells that are each haploid in order to make variety, which can be used to survive environmental changes. So our cell here is considered 2n6, meaning that there's 6 chromosomes total and two parts of the homologous pair, which means that there is paternal and a maternal part for each one. So after the cell goes through interphase and is done work, the cell will be signaled to double each of the chromosomes.
This next step is crossing over, in which the homologous pairs align to make tetrads, and part of this process is the homologous pair sharing genetic information with each other. After crossing over during metaphase 1, homologous pairs can align themselves up in many different ways. Because there are 3 original pairs, the number of combinations is 2 to the power of 3, which means that there are 8 different ways that this can be done.
So this process called independent assortment creates further genetic diversity. Afterwards, homologous pairs are pulled apart into 2 separate cells to create 2 haploid cells with genetic diversity, satisfying 2 requirements for meiosis. So this is the end of meiosis 1, but meiosis is not done yet. These 2 cells contain double the amount of chromosomes than in usable gamete cells, which we are trying to form. And if they fuse, then the resulting baby will have double the amount of chromosomes. So in order to alleviate this, the cells will divide again. Lining up in the middle of the first 2 cells, they will divide to reduce the chromosome number in half. From here, the result is 4 genetically different cells that are haploid. These 4 cells are used in fertilization. So in order to create a baby, for example, these 2 cells will come together and fuse into a new cell that is genetically diverse from its parent. And that's the end of meiosis.